3.2 Carbohydrates. By the end of this section, you will be able to do the following. Discuss the role of carbohydrates in cells and in the extracellular materials of animals and plants. Explain carbohydrate classifications. List common monosaccharides, disaccharides, and polysaccharides. Most people are familiar with carbohydrates, one type of macromolecule, especially when it comes to what we eat. To lose weight, some individuals adhere to low-carb diets. Athletes, in contrast, often carb load before important competitions to ensure that they have enough energy to compete at a high level. Carbohydrates are, in fact, an essential part of our diet. Grains, fruits, and vegetables are all natural carbohydrate sources that provide energy to the body, particularly through glucose, a simple sugar that is a component of starch and an ingredient in many staple foods. Carbohydrates also have other important functions in humans, animals, and plants. Molecular structures. The stoichiometric formula, CH2O, N, where N is the number of carbons in the molecule, represents carbohydrates. In other words, the ratio of carbon to hydrogen to oxygen is 1:2 to 1 in carbohydrate molecules. This formula also explains the origin of the term carbohydrate. The components are carbon, carbo, and the components of water, hence, hydrate. Scientists classify carbohydrates into 3 subtypes: monosaccharides, disaccharides, and polysaccharides. Monosaccharides, mono equals 1, saccharide equals sweet, are simple sugars, the most common of which is glucose. In monosaccharides, the number of carbons usually ranges from 3 to 7. Most monosaccharide names end with the suffix -ose. If the sugar has an aldehyde group, the functional group with the structure R-CHO, it is an aldose, and if it has a ketone group, the functional group with the structure RC equals O-R, it is a ketose. Depending on the number of carbons in the sugar, they can be trioses, 3 carbons, pentoses, 5 carbons, and/or hexoses, 6 carbons. Figure 3.4 illustrates monosaccharides. 3.2 Carbohydrates. 61 Figure 3.4 Scientists classify monosaccharides based on the position of their carbonyl group and the number of carbons in the backbone. Aldoses have a carbonyl group, indicated in green, at the end of the carbon chain, and ketoses have a carbonyl group in the middle of the carbon chain. Trioses, pentoses, and hexoses have 3, 5, and 6 carbon backbones, respectively. The chemical formula for glucose is C6 hydrogen oxide. In humans, glucose is an important source of energy. During cellular respiration, energy releases from glucose, and that energy helps make adenosine triphosphate, ATP. Plants synthesize glucose using carbon dioxide and water, and glucose in turn provides energy requirements for the plant. Humans and other animals that feed on plants often store excess glucose as catabolized, cell breakdown of larger molecules. Starch. Galactose, part of lactose, or milk sugar, and fructose, found in sucrose, in fruit, are other common monosaccharides. Although glucose, galactose, and fructose all have the same chemical formula, C6 hydrogen oxide, they differ structurally and chemically, and are isomers, because of the different arrangement of functional groups around the asymmetric carbon. All these monosaccharides have more than one asymmetric carbon, figure 3.5. 62 Chapter 3, Biological Macromolecules. Access for free at openstax.org. Visual connection. Figure 3.5 Glucose, galactose, and fructose are all hexoses. They are structural isomers, meaning they have the same chemical formula, C6 hydrogen oxide 6, but a different atom arrangement. What kind of sugars are these, aldose or ketose? Glucose, galactose, and fructose are isomeric monosaccharides, hexoses, meaning they have the same chemical formula but have slightly different structures. Glucose and galactose are aldoses, and fructose is a ketose. Monosaccharides can exist as a linear chain or as ring-shaped molecules. In aqueous solutions they are usually in ring forms, figure 3.6. Glucose in a ring form can have 2 different hydroxyl group arrangements. O. Around the anomeric carbon, carbon 1 that becomes asymmetric in the ring formation process. If the hydroxyl group is below carbon number 1 in the sugar, it is in the alpha position, and if it is above the plane, it is in the beta position. 3.2 carbohydrates 63 figure 3.65 and 6 carbon monosaccharides exist in equilibrium between linear and ring forms. When the ring forms, the side chain it closes on locks into an alpha or beta position. Fructose and ribose also form rings, although they form 5-membered rings as opposed to the 6-membered ring of glucose. Disaccharides D equals 2, form when 2 monosaccharides undergo a dehydration reaction, or a condensation reaction or dehydration synthesis. During this process, one monosaccharide's hydroxyl group combines with another monosaccharide's hydrogen, releasing a water molecule and forming a covalent bond. A covalent bond forms between a carbohydrate molecule and another molecule, in this case, between 2 monosaccharides. Scientists call this a glycosidic bond, figure 3.7. Glycosidic bonds, or glycosidic linkages, can be an alpha or beta type. An alpha bond is formed when the O group on the carbon 1 of the first glucose is below the ring plane, and a beta bond is formed when the O group on the carbon 1 is above the ring plane. 64 Chapter 3, Biological Macromolecules access for free at OpenStax.org. Figure 3.7 Sucrose forms when a glucose monomer and a fructose monomer join in a dehydration reaction to form a glycosidic bond. In the process, a water molecule is lost. By convention, the carbon atoms in a monosaccharide are numbered from the terminal carbon closest to the carbonyl group. In sucrose, a glycosidic linkage forms between carbon 1 in glucose and carbon 2 in fructose. Common disaccharides include lactose, maltose, and sucrose, figure 3.8. Lactose is a disaccharide consisting of the monomers glucose and galactose. It is naturally in milk. Maltose, or malt sugar, is a disaccharide formed by a dehydration reaction between 2 glucose molecules. The most common disaccharide is sucrose, or table sugar, which is comprised of glucose and fructose monomers. 3.2. Carbohydrates 65 figure 3.8 Common disaccharides include maltose, grain sugar, lactose, milk sugar, and sucrose, table sugar. Polysaccharides A long chain of monosaccharides linked by glycosidic bonds is a polysaccharide. Poly equals many. The chain may be branched or unbranched, and it may contain different types of monosaccharides. The molecular weight may be 100,000 daltons or more depending on the number of joined monomers. Starch, glycogen, cellulose, and chitin are primary examples of polysaccharides. Plants store starch in the form of sugars. In plants, an amylose and amylopectic mixture, both glucose polymers, comprise these sugars. Plants are able to synthesize glucose, and they store the excess glucose, beyond their immediate energy needs, as starch in different plant parts, including roots and seeds. The starch in the seeds provides food for the embryo as it germinates and can also act as a food source for humans and animals. Enzymes break down the starch that humans consume. For example, an amylase present in saliva catalyzes, or breaks down this starch into smaller molecules, such as maltose and glucose. The cells can then absorb the glucose. Glucose starch comprises monomers that are joined by alpha 1 to 4 or alpha 1 to 6 glycosidic bonds. The numbers 1 to 4 and 1 to 6 refer to the carbon number of the 2 residues that have joined to form the bond. As figure 3.9 illustrates, unbranched glucose monomer chains, only alpha 1 to 4 linkages, form the starch, whereas, amylopectin is a branched polysaccharide, alpha 1 to 6 linkages at the branch points. 66 Chapter 3, Biological Macromolecules access for free at OpenStax.org. Figure 3.9 Amylose and amylopectin are 2 different starch forms. Unbranched glucose monomer chains comprise amylose by alpha 1 to 4 glycosidic linkages. Unbranched glucose monomer chains comprise amylopectin by alpha 1 to 4 and alpha 1 to 6 glycosidic linkages. Because of the way the subunits are joined, the glucose chains have a helical structure. Glycogen, not shown, is similar in structure to amylopectin but more highly branched. Glycogen is the storage form of glucose in humans and other vertebrates and is comprised of monomers of glucose. Glycogen is the animal equivalent of starch and is a highly branched molecule usually stored in liver and muscle cells. Whenever blood glucose levels decrease, glycogen breaks down to release glucose in a process scientists call glycogenolysis. Cellulose is the most abundant natural biopolymer. Cellulose mostly comprises a plant's cell wall. This provides the cell structural support. Wood and paper are mostly cellulosic in nature. Glucose monomers comprise cellulose that beta-1 to 4 glycosidic bonds link, figure 3.10. 3.2. Carbohydrates 67 figure 3.10 in cellulose. Glucose monomers are linked in unbranched chains by beta-1 to 4 glycosidic linkages. Because of the way the glucose subunits are joined, every glucose monomer is flipped relative to the next one, resulting in a linear, fibrous structure. As figure 3.10 shows, every other glucose monomer in cellulose is flipped over, and the monomers are packed tightly as extended long chains. This gives cellulose its rigidity and high tensile strength, which is so important to plant cells. While human digestive enzymes cannot break down the beta-1 to 4 linkage, herbivores such as cows, koalas, and buffaloes are able, with the help of the specialized flora in their stomach, to digest plant material that is rich in cellulose and use it as a food source. In some of these animals, certain species of bacteria and protists reside in the rumen, part of the herbivore's digestive system, and secrete the enzyme cellulase. The appendix of grazing animals also contains bacteria that digest cellulose, giving it an important role in ruminants' digestive systems. Cellulases can break down cellulose into glucose monomers that animals use as an energy source. Termites are also able to break down cellulose because of the presence of other organisms in their bodies that secrete cellulases. Carbohydrates serve various functions in different animals. Arthropods, insects, crustaceans, and others have an outer skeleton, the exoskeleton, which protects their internal body parts, as we see in the bee in figure 3.11. This exoskeleton is made of the biological macromolecule chitin, which is a polysaccharide containing nitrogen. It is made of repeating N-acetyl beta-D-glucosamine units, which are a modified sugar. Chitin is also a major component of fungal cell walls. Fungi are neither animals nor plants and form a kingdom of their own in the domain Eukarya. Figure 3.11. Insects have a hard outer exoskeleton made of chitin, a type of polysaccharide. Credit: Louise Docker, 68 Chapter 3, Biological Macromolecules, access for free at openstax.org. Career Connection. Registered dietitian. Obesity is a worldwide health concern, and many diseases such as diabetes and heart disease are becoming more prevalent because of obesity. This is one of the reasons why people increasingly seek out registered dietitians for advice. Registered dietitians help plan nutrition programs for individuals in various settings. They often work with patients in healthcare facilities, designing nutrition plans to treat and prevent diseases. For example, dietitians may teach a patient with diabetes how to manage blood sugar levels by eating the correct types and amounts of carbohydrates. Dietitians may also work in nursing homes, schools, and private practices. To become a registered dietitian, one needs to earn at least a bachelor's degree in dietetics, nutrition, food technology, or a related field. In addition, registered dietitians must complete a supervised internship program and pass a national exam. Those who pursue careers in dietetics take courses in nutrition, chemistry, biochemistry, biology, microbiology, and human physiology. Dietitians must become experts in the chemistry and physiology, biological functions, of food, proteins, carbohydrates, and fats. Benefits of carbohydrates. Are carbohydrates good for you? Some people believe that carbohydrates are bad and they should avoid them. Some diets completely forbid carbohydrate consumption, claiming that a low-carbohydrate diet helps people to lose weight faster. However, carbohydrates have been an important part of the human diet for thousands of years. Artifacts from ancient civilizations show the presence of wheat, rice, and corn in our ancestors' storage areas. As part of a well-balanced diet, we should supplement carbohydrates with proteins, vitamins, and fats. Calorie-wise, a gram of carbohydrate provides 4.3 kilocalories. For comparison, fats provide 9 kilocalories per gram, a less desirable ratio. Carbohydrates contain soluble and insoluble elements. The insoluble part, fiber, is mostly cellulose. Fiber has many uses. It promotes regular bowel movement by adding bulk, and it regulates the blood glucose consumption rate. Fiber also helps to remove excess cholesterol from the body. Fiber binds to the cholesterol in the small intestine, then attaches to the cholesterol and prevents the cholesterol particles from entering the bloodstream. Cholesterol then exits the body via the feces. Fiber-rich diets also have a protective role in reducing the occurrence of colon cancer. In addition, a meal containing whole grains and vegetables gives a feeling of fullness. As an immediate source of energy, glucose breaks down during the cellular respiration process, which produces ATP, the cell's energy currency. Without consuming carbohydrates, we reduce the availability of instant energy. Eliminating carbohydrates from the diet may be necessary for some people, but such a step may not be healthy for everyone. Link to learning for an additional perspective on carbohydrates. Explore biomolecules. The carbohydrates through this interactive animation. http://openstax.org/l/carbohydrates.